Hello, biology and life science classes. So you guys are actually at the same point in your textbooks as each other. So I'm going to be demonstrating this lab, which is in both of your units in this one video. So we're going to do a blood typing lab, and this is testing type A, B, AB, and O blood types, which are the 4 different blood types. Now each blood type also has an Rh factor, which means it can either be positive or negative. And what does this all mean? So we have type A blood, we have type AB blood, B blood, O blood, we have Rh factors that are negative and positive. What does this all mean? So let me show you here real quick what I have. So I have this lab set up so you can kind of see it a little bit better here. And what I have is I have type A blood. I have a drop of type A, another drop of type A, drop of type B, another drop of type B. This is type AB, and then this is type O. I know it's really hard to see the droplets there, but I'm gonna be explaining as we go so you know what's going on. Okay, whoops, sorry, hands in front of the camera. I'm trying to film myself as I'm doing this lab by myself. So we'll see how this works here. Okay. So what I'm going to start with is explaining the different proteins on your type A, B, AB, and O blood types. So for instance, I guess we should backtrack a little bit. What does the blood type even mean? If you have type A blood, it just means you have a specific protein on your red blood cells. If you have type B blood, that means you have another type of protein on your red blood cells. Now, it doesn't give you like superpowers or anything like that. These are just different types of proteins on your blood cells. Type AB blood has both proteins found on just type A blood and just type B blood. And then type O blood is basically blank. It doesn't have any proteins marking its surface. So what we're going to do today is we're going to use 2 serums. Now, this isn't real blood, obviously, and this isn't real anti-B or anti-A serum because it's not safe to use real blood. So we're just using this as a demonstration, and I'm working with what I've got at home. I didn't have food coloring, so I couldn't dye this red, which is why we are working with milk and vinegar, actually. But type A blood has a specific protein on it. So when we put anti-A serum on type A blood, which we're gonna put this serum on this drop of type A blood, and then we're gonna put, anti-A serum on that one, and then anti-B on this droplet here. And if agglutination occurs, what agglutination is, let me explain that first. So agglutination is basically when your blood coagulates. So if we were to put anti-A serum on type A blood, what should happen? We should get agglutination because that serum is anti-A. So those proteins on our type A blood will react with the anti-A serum, causing agglutination or coagulation. It basically makes it lumpy. If we were to put anti-B serum on type A blood, which we are going to do as well, then agglutination we should predict should not occur because it's anti-B, and type A blood doesn't have anything to do with type B blood, right? Different proteins. So let's go ahead and try that. I'm gonna try to show you guys as I do this. All right. Okay, so the first one's first, we're gonna do, this is a drop of type A blood, another drop of type A blood. So these two are exactly the same type of blood, both type A. Now we're gonna put the anti-A serum on the type A blood. So this is anti-A. I'm gonna try to just do a little drop. Okay, so I'm gonna take this spoon here and just kind of mix it up. And I don't know if you can see this, but it's starting to coagulate. I'll try to zoom in closer so you can see. And that should make sense because we use anti-A serum on type A blood. If it's anti-A, that means it doesn't like A, and it's going to coagulate with it. Now we're gonna put anti-B serum on type A blood. So I'll just do a little drop, as little as I can. Okay, well, that was a lot, but there's no coagulation occurring here. All right, now we're gonna move on to type B blood, and we're gonna use our anti-A serum again on type B blood. These are 2 droplets of type B blood, same exact blood. We'll go ahead and pour that one on there. try to do a little tiny drop. Oh, I keep doing a lot, but oh well. All right, so no coagulation is occurring because it's anti-A. This is type B blood, so it has nothing to do with type B, right? It's anti-A, it doesn't like A. Now we're gonna put anti-B blood on type B, or anti-B serum on type B blood. I know this is kind of confusing, just stick with me. Okay, and if you can see, you can already see coagulation or agglutination has occurred. So anti-B reacted with type B blood. Now I've already done type AB blood, so AB blood we should expect both the anti-A serum and anti-B serum. So both of these serums should react with type AB blood because type AB blood includes both proteins on both type A and type B, so it should react with both of them. Now we're gonna do type O. Type O, the blood droplets are really hard to see as well, but we're gonna go ahead and put anti-A on type O blood, and we should expect nothing to happen because type O doesn't have any proteins on its surface to react. So nothing happened with anti-A serum on type O blood. And nothing happened with anti-B serum on type O blood. So what is our conclusion? Well, our conclusion is that if you have a protein on your blood, whether you have type A blood, type B blood, or type AB blood, you have to have a very specific blood donor if you need a blood transfusion, right? So if I had type A blood, I would need someone to donate blood that also has type A blood. If I had type B blood, I would need someone who has type B blood to donate to me. Now, if I was type AB blood, that means I could accept blood from type A, type B, and type O blood types. So basically, O can be transferable to anyone. They're the universal donor. Type O is the universal donor, meaning they can give their blood to anybody. Now, this is not taking into consideration the Rh factors yet, so keep that in the back of your mind. I'll talk about that in a second. If you have type O blood, meaning you have no proteins, no markers on your red blood cells, that's why you can give it to anybody, because you don't have any proteins that are going to react with their blood type. But if you have, let's do another example. If you have type AB blood, you can accept from everyone. If you have type O blood, you can't accept from everyone. So this is where it gets confusing. People with type O blood, blood like what are they? Like the Red Cross, the associations that take blood. I don't know why that term is escaping me. But if you take blood from people, you really love people with type O blood because you can give their blood to pretty much anybody. If they have O negative blood, they're the universal donor. However, if you have type O blood, you can't accept blood from everyone. You can give blood to everyone, but you can't accept blood from everyone because let's say you do have type O blood. If you accepted blood from someone who has type A, their proteins are gonna react with you because your blood isn't used to having proteins on it, so your body's gonna reject it. And your blood will agglutinate just like you saw with the coagulations here. Not good. That's what happens in your body, and then your body will go into failure. That's why it's really important to get the correct blood transfusion if you have to get one. If you have type O blood and someone with AB blood gives you blood, well, you're going to have agglutination as well. So basically, if you have type O blood, you can give to anybody, but you can't accept from everybody. You can only accept from people with type O blood that have your specific Rh factor. So listen closely. The universal donor is O negative. The universal recipient, the person who can get blood from anybody, but you have to be really careful who they donate to, is AB positive. So people with AB positive blood, they can get blood from people with type A blood, people with type B blood, people with type AB blood, and people with type O blood, no matter if it's positive or negative. So people that are AB positive are the universal recipients. People with type O negative are the universal donors. hope that made sense. I know it's a little bit confusing, but I hope this lab kind of gave you an idea of what would happen to your blood if you got the wrong transfusion. That's why it's so important to get the correct one, and why it's important for healthcare professionals to be doing their jobs and labeling blood correctly, because it would be a huge mistake if they accidentally gave you the wrong blood transfusion. You would die. If you want to try this lab at home, you certainly can. I didn't have all the materials that I needed, but I just used milk and apple cider vinegar. You can use any type of vinegar. Basically, like you need an acid and a base, something that's going to coagulate. So I just decided to use milk and vinegar. And it worked pretty well for the coagulation's sake. But I hope that made sense. I know that's a little confusing. That's why I wanted to do a specific lab on it, even though I didn't have all the materials and couldn't just go out or go to school and grab what I needed. I kind of just put this together with home materials. So I hope this made sense. If you have any questions about blood typing, I think it's really interesting, and I'd love to answer your questions if you had any specific pressing questions on blood types.